Okay, in this video we're going to talk about cell-environment interactions. Now, cells can interact with their environment by responding either directly to other cells through contact or indirectly through extracellular chemical signaling molecules. Now, these interactions between cells always involve this glycocalyx, and the glycocalyx is essentially proteins that protrude out of the extracellular side of the plasma membrane. Now, cell adhesion molecules, or CAMs, are one type of protein you find as a part of the glycocalyx, and the plasma membrane receptors are another. Now, these cell adhesion molecules are basically thousands of these sticky glycoprotein types of membrane proteins that essentially project from the extracellular side of the plasma membrane. Now, glycoprotein is basically a sugary protein. So the protein part is a transmembrane protein in the membrane. The glyco part is basically a sugar that is in a long chain or a polymer, and that sticks out the extracellular side of the membrane, and it can attract to other molecules, which is what gives it its sticky characteristic. Now, functions of these CAMs include things like anchoring the cell to the extracellular matrix. That way these cells can be held in position without floating around within the tissue. They can also assist in the movement of cells past one another, or even attract white blood cells to injured or infected areas. So if tissues get injured, these cell adhesion molecules can help attract white blood cells or immune cells to these injured areas. And cell adhesion molecules can also stimulate the synthesis or degradation of intercellular junctions, like tight junctions, or transmit intracellular signals, like signals within the cell, through migration, proliferation, and specialization. So these play a role in things like development. Now, plasma membrane receptors are essentially proteins that have a binding site on the extracellular side, and these can help with signaling in two major forms. We have contact signaling and chemical signaling. These membrane receptors can actually form direct contacts with other cells, and this is used in normal development as well as immunity. So your immune cells have contact signaling. That way they can help determine self cells from non-self cells, and these non-self cells could be things like foreign microorganisms like bacteria, protozoa, or viruses. And chemical signaling can also be something that can stimulate these plasma membrane receptors, because chemical signaling molecules called ligands can bind to the receptor and then cause changes in activities within the cell, either by triggering enzyme activation within the cell or causing different types of channels to open up that gate ions, and you can change how ions flow across the plasma membrane, which would change the voltage. So some examples of different ligands in the body would be things like neurotransmitters, which is what the brain uses to communicate, hormones, which is what your endocrine glands use to communicate, and paracrines, which is basically what cells use to communicate with other cells within a local area of tissue. Now, what this is showing is an example of a membrane receptor, and it's going to have its binding site on the extracellular side of the receptor. That way these ligands can bind. This could be things like a neurotransmitter, or a hormone, or even a paracrine. And this chemical, once it binds to the receptor site, it causes a change in this receptor that activates something called a G protein. Now, these G proteins are activated once the ligand binds a receptor. It allows for the phosphorylation of GTP onto this G protein, and this activated G protein moves over towards another enzyme called adenylate cyclase. Now, this adenylate cyclase enzyme is something that helps to convert ATP into cyclic AMP, and this cyclic AMP is what we call a second messenger. So what's kind of weird is we started with a chemical messenger outside of the cell. It bound to a receptor that activated a series of molecules, and that activated an enzyme that then caused another activated chemical within the cell. We call this a second messenger because you go from one chemical message to another chemical message, but they started from the same thing. So they're not made of the same thing necessarily, but this activated second messenger, in this example is cyclic AMP, this can go on over and activate different enzymes like protein kinases. Protein kinases are enzymes that actually add a phosphate group onto proteins. And what's interesting about this is that it has a pretty tremendous effect, and it's amplified dramatically. And what these protein kinases do is they go over and they basically help to activate other types of enzymes. They can cause changes in gene activity or gene expression, and the responses are varied. So you can actually even change how cells secrete different molecules. It can change the voltage across a cell. You can even turn on or turn off genes. And this all can be traced back to this ligand binding to a receptor. And so this is an example of an intracellular signaling pathway. And we'll talk about this in future chapters as well.